welcome to the second day of the i2eye meeting. Sure. So that's the first, I was going to say some rules. So first rule, if you're a speaker, take your mask off, that's fine. For those of you who visited the labs this morning, it was really nice to see you all and to welcome you to the labs. For those of you who maybe weren't here yesterday and would like to see us tomorrow, then come and let me know after the meeting today, and we can figure out when you're going to come by. We have two sessions this afternoon, functional imaging and eye movements, first of all, and then we'll have a little pause, and then anterior segment and glaucoma to follow that. And then we're going to end with a session of clinical case share that Michelle's going to animate, where people can show their clinical cases and ask for discussion on different cases they may have. Okay, so I'll maybe pass to the moderator of the first session, Jessica. Maybe I should check that on Zoom everyone is hearing us okay. Yeah, sure, I'll let you do that. I don't know who, do we hear the people on Zoom? Can somebody on Zoom tell us if we're audible?
You're audible. I can hear you.
Thanks, Ethan.
Morning.
Hey. And so yes, on Zoom, if you want to ask a question or if you want to ask a question in the discussion session, feel free to turn on your camera and ask your question. Okay, so join us.
I guess I have to share, right? Could you help me with French? Is it this one? This one here to share?
Yeah, that's it.
Okay.
Oh, Ethan, can you allow us to share the screen?
I don't think
now.
Okay. No, that's all. Where's your presentation? But you're not in full screen.
Yes.
So
yeah.
So you should
put it full screen first.
Okay. Full screen. Then we go to. Yeah, I know, but yes, we should. Double click. And is it? No, it's not. Ouais, c'est plus simple écran. Yeah. Okay. Now they're showing the entire screen, so that's more simple.
Merci.
Just minimize the screen bar.
Okay. Bonjour. C'est un honneur d'être là. It is a huge honor for me to be here today and speak with you. My name is Jessica Morgan, and as many of you know, my lab works in applying adaptive optics imaging technology to study the structure and function of the retina, both in health and disease. And so I want to give you an overview of several of the studies that my lab has been doing recently, in particular with our work on optoretinography, which I'll dive right into. Before I get started, I certainly want to thank the funding sources that have supported this work, in particular, the two grants that we have from the NIH covered this studies from today. And hold on, let me just, since I'm also moderating, I'm going to start a timer on myself as well. And then I also want to just acknowledge the many people who make all this work possible, including all of the folks in my lab, and in particular the three names highlighted here: Rob Cooper, Yu Zhang, and Ray Warner are the individuals that have been responsible for a lot of the work that you'll see today. All right, so I'm going to start by giving an introduction to one of the inherited retinal diseases that we have been studying, called choroideremia. And choroideremia is a rare X-linked inherited retinal degeneration. It's caused by mutations in the CHM gene, and it results in loss of photoreceptors, RPE, and choroid. And if you were to look at a fundus photograph of an individual with choroideremia, this is a typical picture of what you would see. The patient has a retained area of central retina remaining, so the dark area of the center part of the fundus photo, and it's surrounded by regions of atrophy that are ever encroaching inward. And so what my team has been using is adaptive optics to study the structure of the retina within that retained central island. And so as you all are aware here, when you use adaptive optics, you first measure and then correct for the optical aberrations of the eye, such that we can acquire high resolution image of the individual cone photoreceptors. And so along the bottom of this picture is an adaptive optics mosaic acquired with split detection technology. And each little bubble-like feature is an individual cone in the mosaic here. So if I just zoom in, we can look at some regions of interest. And I've just highlighted 3 spots here that show different features that you might observe in your patient with choroideremia. First, in the very center, we have an intact cone mosaic. This is the outline in red, where again, at the top left corner, I've just selected individual cones so you can see where that mosaic looks like. Choroideremia also has these sharp borders of atrophy that abut up right next to the cone mosaic. And so in the orange highlighted area, that the orange dashed line is essentially the area where the photoreceptor mosaic kind of stops and goes off kind of off a cliff. And then beyond that, you no longer have the intact photoreceptors. And then just another feature that's common in choroideremia are these outer retinal tubulations, which on our en-face pictures look like kind of these finger-like projections that extend out from that central area. On the remnant photoreceptors are kind of along that tube. So one of the things that my team has been fortunate to do is actually image a cohort of choroideremia patients that have been treated with an experimental gene therapy. And so we've had 9 patients that have come into our facility. We've been able to image them with adaptive optics, and then they get one eye treated with gene therapy, and the other is left untreated as a control. And so one of the questions we asked is, well, what happens to these photoreceptors following our gene therapy intervention? So here now is an image of the choroideremia subject before and then 1 month following the injection. And I'm just toggling between these 2 images. They're aligned. And what you can see is that following the injection, the mosaic has resettled back on, and the cones are still present after that 1 month time point where they have resettled back onto the RPE following the subretinal injection that lifts the cone mosaic off or makes a retinal detachment. And so we wanted to quantify these results, and so we selected a number of regions of interest in all 9 of our subjects, both in their injected and control eyes at both time points, and measured cone density over these regions of interest in all of those cases. And so here now is plotted cone densities at the 1 month time point versus cone densities at the baseline. The green is the injected eyes, the gray triangles are the control eyes. And what you see is that the cone density remains stable in the 1 month post-injection time point. So we found no statistical difference in cone density across time points or between the injected or uninjected eye. And this leads to 2 really important safety conclusions related to the gene therapy intervention. Was first, that the surgical intervention itself, the retinal detachment that's caused by the surgery, did not cause widespread cone loss in these individuals. And that second, the study agent itself did not cause an acute toxicity. And so these are results that we've just recently published earlier this year in JAMA Ophthalmology. And so that's fantastic. It's been really rewarding to be involved in imaging this cohort of individuals. I really consider them part of our study team. They're very invested and in coming back and doing all of the many things that we ask them to do. But what I've told you about so far is really about safety. And so one of the things that we want to know is, well, is the gene therapy working? What about efficacy? And so what I want to impress upon you is that just because we see a photoreceptor is visible in the image, that doesn't necessarily mean that the cell is functional. And so we need to develop methods that can assess the function of cells at the same time that we're also assessing their structure. And when I think about, in particular, relating things to choroideremia is really tough to have an efficacy endpoint because the subjects start with high acuity and they maintain that until very late in the disease process. And so trying to find a way that would show either an improvement it can be difficult. And so many of the endpoints for clinical trials are actually designed to show halting of progression. And so it would be wonderful if we had additional techniques to investigate cellular function. So with that, I'm going to dive into a technique that we've been studying, which is optoretinography. And optoretinography is a technique for measuring an optical signal that arises from the retina in response to a visible stimulus. And the corollary is the well-known electroretinography, or ERG, where we measure the electrical signal that comes from a visible stimulus. And optoretinography is not a new idea. In fact, it was the first publication of it came in 1994 at an OSA meeting. And at that time, though, Jeff Mulligan and Don McLeod were unable to measure an optoretinogram signal. But the idea that you could get an optical signal in response to the visible stimulus has been around for quite some time. And then more recently, with the invention of adaptive optics and the combination of adaptive optics, more and more of these studies have popped up over the years. Over the years in terms of trying to find this optical signal, or the optoretinogram. So my team uses adaptive optics scanning laser ophthalmoscopy. And so I'm going to describe the results that we get using that technique for this optoretinography idea. So this is a video here. I really hope it's playing well through Zoom for all the folks online. But on the right, we have a confocal AOSLO image sequence. It's acquired with near infrared imaging source. And we've stabilized, done all the registration so the retina remains stable in the image. And then while imaging with our near infrared source, we apply a visible stimulus to the entire imaging field. And we do that through the raster scan. So in this case, it's a red stimulus that we've applied to that whole field. And then the idea is to essentially watch what happens to the near infrared reflectance following the stimulation. So I've identified one cone outlined in the orange here, and we plot their reflectance trace below it on the left. And so our cone starts with an initial reflectance in the pre-stimulus case. The light turns on, and this cone's reflectance increases and then decreases in response to the light. And then if I show a second cone from the same video, what we see is that this cone also starts with an initial reflectance. The visible stimulus comes on, and then this cone decreases its reflectance. And then if I pick still a third cone, here we have even a third reflectance pattern that happens. So the cone, again, starts with its initial reflectance value. The light comes on, and then its reflectance oscillates in response to that visible stimulus. So what I want to impress upon you is that the cone reflectance change in the near infrared is highly heterogeneous. And in fact, if you looked at any one of the any one of the many cones in this picture here, you would get a unique reflectance trace in response to that visible stimulus. So we wanted to ask, to what extent is this change in near-infrared reflectance following the visible stimulus an optical biomarker of cone function? Or in other words, can this change in near-infrared reflectance be an optoretinogram? And so the first thing we had to do was find a way to quantify this signal. We spent a long time on this because the signal was so heterogeneous between cones. What we settled on is a technique that's fairly well used in neuroscience, which is to standardize the cone's behavior. And so we standardize the reflectance for each cone across the image, meaning that we give the overall ensemble of cones a mean of 0 and a standard deviation of 1 in the pre-stimulus condition. And then when the light comes on, we then watch how the cone's reflectance diverges from its initial value. And so this video here shows the pre-stimulus condition kind of as like the more gray, uniform gray appearance. And then the light comes on and you see the cones diverge and get either bright or dark in comparison to their pre-stimulus reflectance behavior. And so then to quantify this further, we actually take, we just plot each cone's essentially standard deviation, their standardized reflectance over here on the right. And so this now has a red line or red trace for each individual cone in the image. And you can see it starts off with that standardized value in the pre-stimulus condition. And then when the light turns on, the cones diverge. And so to quantify this down to then a single signal, we've taken this population level of cones, and we simply take the standard deviation now of all of those traces across the response, and this now becomes one of the signals that we're looking at. And we can take essentially that heterogeneous set of cone responses and boil it down to one graph that has properties like this, all right? And so we've done this both in in stimulated and control, like non-stimulated conditions. And we simply just subtract off the control, and now we end up with a trace like this for a signal where we've stimulated a population of cones. And so from this point on in the talk, I will call this red trace here our optoretinogram, or the ORG. All right. So then we wanted to know, all right, now that we have a way to quantify this result, we wanted to ask some basic properties about it in relation to the visual system. So for a response to be a functional biomarker, we would need to have essentially a dose response. So if we stimulated with dim lights, we would expect a different level of response than if we stimulated with a bright light. And so we did that experiment. We tested with a range of irradiances for our stimulus. And so here are the different traces plotted. And essentially what you see is as we increase the light level of the stimulus, we get an increasing response of the optoretinogram. And if I do this now for a number of subjects, and we just plot now the amplitude, so how high the trace goes on the y-axis here, we can see this, again, dose response in all of the subjects that we tested. And so this is, again, maybe the first real evidence that our signal does in fact follow properties of the visual system and may serve as a biomarker for cone function. So we wanted to further test that, and we did this by varying the wavelength of the stimuli in order to see what is the action spectrum, what is the spectrum that this type of response would follow. And so if we now test over a range of wavelengths, we can measure the action spectrum, which is these the orange points plotted there. And we find that the action spectrum of this optoretinogram signal corresponds very nicely with the human photopic luminosity function, or in other words, the spectral sensitivity of the eye. And so this then becomes, again, really strong evidence that the optoretinogram that we're measuring across the population is a biomarker then of our cone vision or cone function. We published these results several years ago now in Biomedical Optics Express. So in order to now take this signal and start applying it in cases of disease or translating to more clinically relevant studies, we have to understand properties of the signal. And in particular, how much is this optoretinogram signal repeatable? Because this is really important for putting error bars on any of the studies that we're going to do moving forward into the clinic. And so we designed an experiment that would let us test or the repeatability both within and across sessions, and then also the repeatability between stimuli of equal energy conditions. And so here's a trace from a single subject using, I guess this is in session 1, but like just one part of the data in session 1. And so we then repeated the exact experiment within that time. In fact, it was actually randomized throughout the imaging session. And so if we look at the same condition here, we see the same trace. And so this lets us know that amplitudes, the ORG amplitude was repeatable within a session for this subject, which is great. So we combined that data into one condition here. And then we said, all right, well, what about if we tested equal energy stimuli? So maybe a bright flash for a short amount of time in comparison with a dimmer flash that extends longer for a longer duration. And so this is a condition now where we had the dimmer flash, but for a longer period of time. And what you see is that the amplitude builds to about the same height, but that it takes longer to do that. The slope or the impulse part of when that optoretinogram builds up takes a little bit longer in the dim light condition, which makes sense when you think about how the cones are receiving that light. So then we tested a couple of different durations in between this range. And I'll just bring them up here, and the same trend holds. Essentially, all these equal energy stimulus conditions produced amplitudes that were basically the same with varying slope depending on the duration tested. So then we said, okay, that this is really important information for us to know as we design studies that can go into clinical populations and where we can also make stimulus conditions that are comfortable for viewing. And so then we repeated the entire experiment a second time. And so here is that data, again, essentially showing that all of these amplitudes were also repeatable across the sessions. So we did this in a number of subjects. So I'm just showing you the summary data here. And the trends continued to hold. So we found, again, no significant difference in amplitude within a single session and no difference between the sessions. What we did find, however, and this is very interesting, is that there were differences between individual subjects. And so if you look at the numbers on the left or the y-axis of these plots, you'll see from the different subjects, we have 5 here, that the amplitudes that those subjects reached were varied between the subjects. And so I took their amplitudes and I've plotted now the amplitude per subject for session 1 versus session 2 on this plot down here on the bottom right. And essentially what this is showing is that the variability for a single subject, the error bars on those red dots, are small in comparison to the variability between subjects, which is essentially the difference between the distance on the dotted line there. And so this leads to more questions that we have in terms of understanding population level norms and what might be causing these individual variations between subjects. All of these individuals tested here are normal sighted controls. All right, so now I want to take a step back again. And when we first started talking about the optoretinogram, I showed you that the reflectance following the stimulus for these cones was highly heterogeneous across cones. And so this is now the optoretinogram that I've talked about so far has been a cone level measurement that's aggregated across a population of cells, right? But when we start thinking about identifying cells that have, individual cells, for instance, that have responded to a gene therapy, we want a measure that's on the resolution of an individual cell. And so we've begun to translate our population level analyses into a single cone optoretinogram measurement. And so the thing to know in order to do that is that the signals that we get from an individual cone are also highly heterogeneous across trials. So if you stimulate the cone in one trial and measure its reflectance, you'll get one of these traces here that you see on the right side of the graph in red. But if you repeat the same stimulus on another trial, that cone's reflectance pattern will be, again, unique. And so instead of now aggregating signals across cones within a single trial, we can aggregate signals across trials from the same cone. And in that way, still use essentially the same mathematics in the background to pull out a single cone optoretinogram response. And so we have done this, and we can now repeat, multiple trials in rapid succession, acquiring these image sequences, pull out the cone reflectance trace for each of those trials, align them up and aggregate across trials, and essentially come up with an optoretinogram amplitude for each individual cone. And so shown here now is a pseudocolored mosaic based upon the cone's optoretinogram response. And without going into the details, but I'd be happy to talk about them afterwards offline, the cones that are outlined in white, we have identified as S cones based upon having a low optoretinogram response to our 550 nanometer stimuli. All right, so now we want to translate this back to our disease cases. And so we've begun looking at this in choroideremia, and these are very preliminary results. But we have 4 choroideremia subjects that we've imaged in the paraphobia in comparison with our 5 controls that are, again, eccentricity matched. And when we now look at, again, the population level analysis, we can summarize their optoretinogram here, showing that the cones in the choroideremia subjects, although structurally they appear relatively normal looking, and they are in near normal numbers, the optoretinogram amplitude is reduced in these subjects in comparison to the normal-sided controls. And so with that, I'll just summarize that their cone optoretinogram is measurable across a population of cones and at the individual level, and that it shows high potential as an objective biomarker for assessing cone function both in health and disease. So thank you very much. Merci. I'm happy to take your questions. Laura.
So you said that for a single cone, the response changes from trial to trial. I just wondering what the source of that is.
Sure. So the results that are coming from adaptive optics, OCT, and optoretinography suggest that there is an elongation in the cone outer segment that occurs in response to the visible stimulus. So how that translates to the SLO images is really a matter of coherence. So our SLO images don't have the actual axial sectioning that OCT has. And so the image is a combination of the cone outer segment tip and the ISOS junction. You can imagine those reflectances kind of superimposed together. So depending on the initial distance between those 2 points, if we stimulate with light and you imagine the cone getting slightly longer, right? But then those 2 reflectances still being superimposed. Sometimes you might have constructive interference between them, and other times destructive interference between them. And so we believe that this is what causes the heterogeneity of the signal. So just depending on what that initial length is and how much it changes, you might have some cones reflectances that go up or down, and that gives the kind of a cross cone response. But in the same way, when you think about across trials for a single cone, the initial starting point might be different trial to trial, and therefore whether you're getting constructive or destructive interference trial to trial might also change. Yes.
Actually, I have a follow-up question. Do you know if in AOCP that they didn't find this heterogeneity in controls, or did they also find
Okay, so I'll try to repeat for the folks on Zoom. The question is in the OCT work, did they find heterogeneity across controls? So I think the OCT signals, when you're looking on repeated trials, their traces are come out a bit different because they're measuring a change in the phase. And so the OCT community is seeing an initial contraction in phase followed by an elongation. That seems to be fairly consistent. I don't know if it has been shown, or if it has, I'm not aware of the results that would look at the end amplitudes that they get across controls. Not sure there. Yes.
how do you stimulate phones, like one by one, or like a few of them? Because they you showed the for 2 seconds.
Sure. So, okay, the question is, how did we stimulate the cones? Were they one by one or in the population, and for the duration? So the stimulation is actually provided through the adaptive optics system. So the stimuli or the stimulus is also scanned through the entire raster pattern. So it does cover the entire field of view. And we would essentially, we turn the light on and leave it on for 2 seconds, but during that 2-second time, it is scanned over the full field, and then have a shutter that turns it off. And 2 seconds was one of the durations that we used regularly in the initial work, but we've actually now been shortening that stimuli. It's a lot more pleasant and easy to acquire the data if we just do a much shorter duration, essentially just one raster pattern scan, a little quicker. Other questions?
Can I ask a question?
Yes.
You said there was some difference in amplitudes between the normals. Was there any age-related or fraction-related difference?
So that's a great question. And the results that I showed are in 5 subjects. So, I have to take it with a little bit of a grain of salt there. We do have a broader population level study planned where we'll be looking at age and then also sex and race. So it's difficult to say. The 5 subjects that were there did range in the lab participants, right? So your 20 to 45-year-old group.
So what was the question again? Because
Oh, I'm sorry.
Age-related or refraction-related?
Yeah. Sorry, for Zoom, it was the differences between controls related to age. Yes.
Not a question, question on the.
Oh, a question on Zoom. All right, I apologize. I apologize to the Zoom folks here. Let me pull it up. Okay, so here we go. Was it necessary in your work to image subjects at the same time of day across trials, given circadian linked pattern of photoreceptor function? Fantastic question. Yes, we did control for the time of day. In fact, in our repeatability studies, we required imaging in the 2 different sessions to happen at the same time of day. Of course, this leads to questions about whether or not the optoretinogram signal would vary across time, and we do not yet know that. So good question. And have we compared ERG signals between these patients and control subjects? Oh, well, it's also a really good question. We really want to do this because the signals, it begs to be done in terms of comparing, in particular, the A wave of the ERG with the optoretinogram measurements. We haven't done it yet. It does become a little difficult when you start thinking about this. ERG, a classic ERG will sum up signals across the entire full field of the retina. So for instance, our choroideremia subjects, they'll just have a flat ERG. They don't respond on it because their central island is so small in comparison to the periphery, which is just, full loss of photoreceptors. So we want to do that. We will have to do it in like a multifocal ERG setting for sure. Yes.
I have interest to see if you have already had the carriers, like female carriers in the choroideremia communities, because when they see the family, the RPE as so in the periphery, like aged people, they show the various the features, but they have no symptom. So
Right.
I really want to see if you already done in AOSL or functional.
Right. We haven't done the functional studies in those individuals yet. We do have some AO images of them. As you say, there is a phenotype there, but perception-wise, the carriers don't observe too much. I've been really approaching this from the gene therapy side. We've been very heavily invested in this group of gene therapy. And the phenotype of the carriers actually lets us think a lot about how good does your therapy need to be, right? So in choroideremia, the carriers that you would think on average have 50% of their cells affected do not have a phenotype. And so, if you could show that your treatment was effective in 50% of the cells of your patients, that might be sufficient. And so, when you think about that, then that provides even more motivation for really wanting a cellular level assessment of intervention efficacy. All right, I'm going to cut myself off because I have to moderate the rest of this session, but I'm happy to take additional questions in the discussion time. It really was my honor to be here and talk with you about this today. With that, we'll go to the next talk for the session, which is Emiliano. I'll let you pull up your. Yeah.
Good morning, everybody. My name is Emiliano Ronzitti. I'm a researcher at the Instituto della Visione, not so far. So what I'm gonna talk today is a project that we recently carried at the lab, where we developed an all-optical system to study retinal circuits into the mouse retina. So we all know that retina is an outpost of the brain for visual scene processing. Very briefly, we have the photoreceptors, detect light. The signal is passed to a number of inner microcircuits that involve bipolar cells, amacrine cells, and others, releasing information to the retinal ganglion cells that then send this information to the brain. And there are a number of microcircuits that works in parallel, such that different feature of the you can take it to the microphone. What? No. Yeah, I was saying, so there are several microcircuits that works in parallel, such that different feature of the visual scene are processed, and we have multiple representation of the visual scene to our brain. Now, how do we study these microcircuits? More in general, how do we study neuronal circuits? There are obviously several approaches. I would say that we can group them in two microfamilies. So we have some imaging approach. Here, the purpose is to identify the structures morphology, the connections, so studying from where the signal is coming from. And on the other side, we have electrophysiology methods. In this case, we can study when the signal appears, and so following the dynamics. And to a certain extent, we can both monitor and manipulate a bit the dynamics of those circuits. Now, quite in the last couple of decades, a series of techniques have been developed, so-called optogenetics tools, that in a way can fuse a bit together, can merge a bit together, few benefits of those macro areas. So we have some, on one side, some sensor, some fluorescence, which are sensitive to the activity of the neurons, like a calcium indicator or voltage indicators, such that we can translate some neuronal activity to some fluorescent transients. And we can genetically express those tools into the neurons in the specific cell type. In the very same moment, we can also express some light-sensitive actuators, some microbial opsins can be expressed to some specific cell type. And so we have a couple of tools, thanks to which we can monitor and control the neuronal activity. And so, for example, we can find out which is the involvement of a certain cell type in a certain task, in a certain behavior, or in a certain pathology. Now, usually, these kind of approaches presents a wide field illumination, as the one that we can see here. So what happens is that we can monitor the activity of the neurons, but when we want to activate them, we light up all together. But if we look to what happens spontaneously in a network, it is something quite different. I mean, not all the neurons light up together. We have a certain neuronal code that in the most of the cases is what we wanted to study. Or sometimes we have cells which are functionally different, but genetically similar, like, for example, the direction selective cells. So in the last years, we developed a series of approaches which use a special light modulator, a 2-photon illumination beam, and the phase modulation of the incoming light, such that then we can rearrange the light, we can shape our light just to target it on some specific location, such that we can track back and eventually play back a certain activity. And this number of techniques, they are usually called in the field all-optical methods to read and write on the neural circuits. So we applied recently those techniques in mouse visual cortex, and we could indeed track back a certain neuronal activity in selected targeted cells. So, for example, in this case, we patched a cell in the visual cortex of a mouse. We record its activity, this is the black line, and we could play back this activity. We can induce back this activity by sending light on these cells. And we can do that at the level of the single cells, and we could do that at the level of multiple cells with different degrees of precision, where we can claim to have a near single cell resolution of stimulation. And so the idea was to apply those techniques to study the retinal circuits. Retinal circuits present some advantage compared to the brain circuits because the scattering is pretty low. It presents some challenges because the signal processing evolve in a direction which is parallel to the propagation direction of the light. And when we use focused light, then it becomes quite challenging to maintain a certain axial resolution. So the goal that we wanted, it was that we wanted to develop a unique all-optical system in which we could control the input of the retina. So we wanted to control the light at the photoreceptors. We wanted to monitor the widest population of ganglion cells simultaneously. And then we wanted to have the possibility to activate selectively some cells in the inner sequence. And so we developed the following system. I don't enter into the details of that. I just wanted to describe the main units of this system. It is obviously a multi-unit system, and the main units are devoted to these tasks that I was saying at the beginning. So concerning the possibility to monitor the output activity of the retina, we built up a system which is a 2-photon scanning microscope, which is pretty basic. We have a titanium sapphire laser, which is projected onto some galvanometric mirror, such that we can steer the beam on our field of view. And then we express a calcium indicator selectively in the ganglion cells with intravitreal IAV2 injection, and we could follow the dynamics of the retinal population with at about 6-10 rate. And at the very same moment, we could produce some visual stimulus on the photoreceptors by second unit here at the bottom, where we built up a system based on some digital micromirror device. In this case, we use some visible light provided by an LED, and we could produce different kind of shapes. So, for instance, in this case, we could produce some moving bars, and then we could play with the two together, and then we could, for example, classify different kind of ganglion cells. So, for example, in this case, we classified 4 different kind of direction selectives. Now, concerning the light targeting inner circuits, this system is a bit more complex. It's less also the rest is complex, but this is more complex, and it is a bit less conventional. We can identify 3 main stations to shape the illumination. The beginning, we have a phase mask, a fixed phase mask that produce a phase modulation in a plane which is conjugated to the back focal plane of an objective lens. In this way, by producing a proper phase modulation, you can shape the light with the shape that you want, a star, a disk. In most of the cases, is a disk that is roughly the size of the cell. Then we have specialized modulators, which is in a following conjugated plane to the phase mask, to the back aperture of the objective. And in this case here, we can encode another phase modulation that encode for some diffraction limited spot in the field of excitation. So the effect of these 2 steps modulation is basically that you produce a certain shape, a star or a disk, and you can multiplex it in different positions 3-dimensionally in your field of view. Then we have a third ingredient, because we wanted to have a pretty high axial resolution, and we use a technique which is called temporal focusing. I don't want to enter too much into the details, but the point is that when you have a pulsed beam and you disperse it by using a kind of grating, then you can create an effect such that the pulses becomes quite stretched in all the outer focus plane, while they becomes quite narrow in time at the focal plane. What happens is that you have a very high density of photons when you are in the focal plane of your interest, and the photons are quite diluted in all the outer focus plane. So basically, you create a 2-photon excitation only in a certain location. In a certain location of focal plane. Okay, so with these three ingredients, we could produce some light-targeted spots. Here is an example in volumes which are of about 200 by 200, a few hundreds micron laterally, and a few hundreds micron axially. In this case, remember it is 300 by 300 by 500 micron. And so we tested the capability of the systems to study the range to which we were interested. So from photoreceptors to ganglion cells in the mouse retina, we are about 100 micron. So we wanted to test if we were able to move these spots precisely across these 100 micron by keeping a certain XYZ resolution, and that was the case. The lateral resolution was pretty well maintained at about 10 micron, which was the spot size that we encoded, and also the axial resolution as fluidal maximum was at about 10 micron. And we checked that in all the field of excitation, we had quite an homogeneous, spots in terms of, again, axial resolution, axial and lateral resolution, and intensity. Okay, then we started to test that to activate some bipolar cells, which were treated to express microbial opsin, a COCHR, to become sensitive to light. And we started to see how good it was, our light target. Just a couple of words on these, on the bipolar cells that have been targeted. In this case, we decided to target broad bipolar cells. That was in the framework of a collaboration with Olivier Marre and Serge Picaud at the Institut de la Vision. And actually, the pathway of the broad bipolar cells is a pretty challenging pathway in terms of studies of its functionality, because it's a pretty divergent pathway in the mouse retina, which is polysynaptic. Rod bipolar cells communicate with many cone cells, so it is pretty difficult to study with anatomical approaches or electrophysiological approaches, and such divergency pose some questions about the level of resolution, spatial resolution that could maintain these kinds of pathway. So we started to use our methods to targeting a few rod bipolar cells. And so for doing that, we use patch clamp, and so we patch one positive cells, having removed the photoreceptors, and then we started to move our our illumination spot axially and laterally to see what was the precision of our photostimulation. At the beginning, we verified that we could produce responses in the rod bipolar cells that were comparable with the physiological ones, and that was the case for certain light intensity. And at this light intensity, we started to play and to see which was our resolution. And the lateral resolution, so here I'm plotting some the responses of the cells when we illuminate it, so-called photocurrent, because they are current which are induced upon illumination. And if we consider the fluidal maximum of this bell-like profile, we obtain a lateral resolution which is roughly the dimension of the cell, so around 10-15 micron. Actually, we are a bit more dirty. In this case, we are at around 30 micron, but it is good enough to be close to single cell resolution. And then we started to play with all what we had. So the idea was to start to see what happened when we were photostimulating selectively some rhodopipular cells while we were looking at the reaction of the ganglion cells. And we found that we could induce different kind of responses in the ganglion cells by using different patterns of illumination of the rhodopipular cells. We used groups of spots between 2 and 10 spots, and we could identify that also not so high variation in the distribution of the spots could induce a significant difference in the responses of the ganglion cells. And importantly, we had to conduct these experiments in presence of a pharmacological cocktail that at a certain moment cut the transmission between photoreceptors and bipolar cells, because unfortunately, at a certain moment we realized that even if we were in the infrared, probably the fact that we are quite far infrared, even if we were quite axially confined, we could have massive responses from the photoreceptor. So we are sure that these responses, they are just induced by the rhodopipular cells activation and not by the photoreceptors. And so we did many different trials in which we produced different patterns of activation of the rhodopipular cells, and then we put together the data and we modeled a bit what we observed. And what we found out, it was that the response of the ganglion cells, they were quite confined laterally to the position of the rhodopipular cells. So we studied so-called cellular receptive field, which we defined as the area across which ganglion cells respond with the photoactivation of the rhodopipular cells. And we confirm that this cellular receptive field has an extension of few tens of microns, which is compatible with classic receptive fields, and that confirm that these kind of pathways, the rhodopipular pathway, even if it is so divergent, could maintain an high resolution picture of the high resolution, especially high resolution picture. And we confirm that on different cell types. As I told you before, we could classify different ganglion cells by using different stimulation with digital micromirror device. And we confirm these results, so this confinement of the cellular receptive fields, and so this high resolution possibility of the rhodopipular cell pathway. So with that, I concluded. So what we developed so far, it is a system that can use light to study neural circuits into the mouse retina. We could monitor the retina output with calcium imaging and 2-photon scanning. We could play with the visual inputs with visible light digital micromirror devices, and we can manipulate selectively at single cell level the inner circuits to see the involvements of different cell types of the different single cells. And in particular, in the framework of this project, and we studied particularly the rhodopipular cell pathway, which is particularly divergent and pretty difficult to be studied with the traditional methods. And so I concluded, obviously, that is the work of a team. It was a fruitful collaboration with many people at the Institut de la Vision, especially the group of Olivier Marre and Serge Picaud. and the group of the Dalkara for the expression, the selective expression of the microbial opsin and the GCAM indicator, and of our laboratory at the photonics department of the Institute of PCO. So thanks for the attention.
There's time for questions. Veronika, do you want to get set up to start while we? Question? Fascinating work. Will you be able to translate into disease models where the photoreceptors have are where if the photoreceptors are degenerated in your model, will you be able to recapitulate signals?
In mouse retina, you mean, or I mean the system is pretty flexible, so it can be obviously applied to different kind of situation, also in pathological states. So yeah, I don't see if it would be yeah, it would be definitely possible.
Fascinating. All right, well, thank your speaker one more time. Time for one more question if you have. Did you have one?
Oh, yeah. Just a quick question. In your multiplexing setup, where you use both the faceplate and spatial light modulator,
yeah.
Why did you choose to include everything in the hologram displayed on
excellent
the
excellent question. Actually, that is the traditional way to do that. The problem is that when you want to maintain a pretty high axial confinement for this focusing beam, then and you want to have relatively extended spot, the dimension of the cell, then pretty quickly the axial profile is deteriorated. So you have to use this temporal focusing. And actually this temporal focusing breaks somehow the possibility of doing a single phase modulation of all together. So we found out, we figured out this intermediate strategy of the 2 steps phase modulation. But if you don't want to maintain this confinement, definitely possible to do direct 1 phase step.
So this is a short comment. Yes, your technology may help to answer some of the question of your talk about the pattern of the reflective observability you go on the photoreceptors. Just something very exciting to verify.
I was thinking to that, and actually, yeah, that is something that to be discussed. Thanks.
Thank you again. And our next speaker is Veronika. All set up. Go ahead.
Thank you. Hello, everyone. I'm from Bonn Adaptive Optics Lab, and today I will show you some of our results on the influence of the drift into visual acuity. And so we all know that while we fixate steadily, image which falls into the eye actually moves across a number of photoreceptors. And on this video, you can see, okay, it's mouse, but you can see all of the fixational eye motion, and fixational eye motion includes usually 3 components, which is random light drift, high frequency tremor, and microsaccades, which are rapidly changing gaze to the optimal retinal location. And so vision is an active process, and drift produces transient visual signals. And as you can see on this video, sorry, it's complicated. So as you can see on this video, while subject perform an acuity task, you can see some cone activation patterns. And so we know that probably due to fixational eye motion, specifically drift, like some information is adding out during the presentation of the stimulus. And so what is also known is that people are extremely good in performing visual acuity tasks. And so here we can see different observers. This is work from Rossi et al. And people are extremely good at doing acuity tasks. Sometimes it can go beyond even the nyquist limit or the cone resolution. And so one may ask here, what is the role specifically of the drift in such a good visual acuity performance? And so it's been shown by Michele Rucci and his lab that actually drift enhanced visual acuity. And so how they showed it, they simply stabilized fixational eye motion. So they compared for performance here on y-axis when image was like just presented naturally, or when it was stabilized in the retina, or here stabilized is red. And so what we see is that when image is stabilized in the retina, performance drops. And so in our experiments, we also wanted to check how drift influence visual acuity, and more specifically, we ask ourselves the question of how much drift do we need to reach maximum acuity performance. And already here you can see that when we just normally observe image, it travels quite a lot in the retina, but when we stabilize, it activates relatively small retinal area. And so we are going to address this question throughout my talk of how much of the drift do we need to reach maximum performance. And so for our experiments, we use adaptive optics scanning laser microsimulation system. And all the experiments which I will show here, they've been performed on a very central retina with the field of view of 0.85 degrees of the visual angle, or like 512 pixel raster size. And in our case, raster is actually a display where we present something for the subject. And per 1 second, we have 27 frames. So it means that roughly to scan 1 frame, it takes 37 milliseconds for us. And in these experiments, we used 788 nanometer wavelengths, and all the subjects, they've been imaged monocularly on a dilated pupil. And so to test acuity, we use just a simple Snell and E acuity target, and here I present data for 4 participants. And so it's been shown before here on y-axis that performance with increasing exposure duration on x-axis actually getting better in terms of visual acuity. So we thought that it might be due to increasing fixational drift. And so we wanted to manipulate also in our experiments, manipulate exposure duration in order to increase linear drift amplitude. And so linear drift amplitude we measured as a concatenated vector length of the drift motion during the stimulus presentation. So we had like 5 presentation duration intervals in our experiments, and we actually achieved it. So linear drift amplitude grew proportionally to the exposure duration, which is of course expected. And so here you can see example of a typical trial in our experiment. So stimulus is present in the middle of the raster, and person observes, and retina moves on the background. And here you can see drift traces for 23 trials for 1 participant. And so we can see clearly that in a natural eye motion condition, stimulus moves quite a lot across the retina. And so again, linear drift amplitude increased, but what would happen with the area on the retina? And so here with this light gray, you can see that actually the retina also grew, and how we measured like this area of the retina, we measured it with the drift semi-perimeter of the motion trace. And so it also proportionally grew towards the increasing exposure duration of the stimulus. But what will happen in stabilized condition? And so retinally stabilized, meaning that something moving together with the retina. So basically what we did, we measured preferred retinal location of each participant, and then placed continuously stimulus in the same retinal location. And so you can see it's kind of glued to the retina and moves together with the retina. And so here you can see drift traces again for 23 trials, but in stabilized condition. And what we observe here is actually that our stabilization is not perfect. So there is still some residual jitter or slip, we call it. But area of the retina is very restricted. And so this linear drift amplitude also grew together with even in stabilized condition, but was reduced compared to the normal eye motion condition by the factor of 1.6. And what will happen with semi-drift of the semi-perimeter of the drift? Actually, it didn't grow much, and it was decreased in stabilized condition by the factor of 5.2 for the longest presentation duration. And in our systems, our main advantage is that we can resolve each individual cone on the fovea. And so here we plotted foveal profiles for 4 participants. And we got one participant with a relatively low cone density, and it's centered across the cone density centroid, and some participants which are like on the more higher end of the cone density. And so here I plotted some parts of the retina for 4 participants. And so we can see that for participants with the lowest cone density, we can actually see each individual cone perfectly, but with increasing cone density, our resolution drops a bit, even so we still can resolve and see each individual cone. And so we wanted to check how actually cone topography influence drift behavior. And so here I plotted some drift traces for different participants, just some random drift traces. And so what is interesting is this participant with the lowest cone density on average did all the same type of motion, more elongated motion, even though some participants sometimes will also do this elongated type of motion, but more rarely. So on this slide, I plotted again for 4 participants some parts of the retina across a cone density centroid. And this subject is of particular interest. So she's completely healthy, a healthy person, but we can see that she continuously doing the same type of stereotypical eye motion. So she will start somewhere on the upper left corner of the retina and then move stimulus with natural eye motion towards the cone density centroid. With other participants, it was not the case, so they usually would do some more or less random motion around the most like dense area. And so we compared cone topography to the threshold or performance, visual acuity performance, and again we see that participants with the lowest cone density on average performed worse compared to the people with higher cone density. And it's been shown by a PhD student in our lab, Ginny Witten, that actually, yes, there is a correlation between the cone density and acuity threshold. And so the higher the cone density, the lower the threshold. And this work is preparation, so stay tuned. And so we compared also some of the other drift behavior. So here I plotted drift velocity, and we can see again that for the subject with the lowest cone density, actually drift velocity was on average a bit bigger, but compared to, like, for example, participant number 2, but we in a way still see this trend with decreasing drift velocity with increasing cone density. But what's interesting with this subject is the variation of drift velocity was much higher. And then linear drift amplitude, and it's here plotted only for the longest presentation duration, as here traces plotted also for longer presentation duration. And so we can see again that a linear drift amplitude for her on average been higher and variability also bigger. So here we conclude that cone topography actually influenced drift behavior. And so coming to the analysis of the acuity performance, so here you can see 4 plots for 4 participants, and I plotted here natural eye motion condition and stabilized. They are together here. So natural is filled, stabilized is empty boxes. And so what we can see here is that clearly all the participants, they performed worse for very brief flash, like just for 2 milliseconds when like stimulus been flashing. But then like almost everybody reached a plateau. And what we clearly didn't see here, a significant difference between stabilized condition and natural eye motion condition, which is of course a surprise. And so here I just plotted threshold, so it's clearly, we can clearly see that there is no difference within the subject, but between the subject, again, there is like a clear trend of decreasing threshold with increasing cone density. And so what we decided to do, actually completely reshuffle the data and look at it from another perspective. So here we actually sorted all the motion traces according to the linear drift amplitude in arc minutes on the x-axis, and for 4 different participants. And so we can see that like this plateau of maximum acuity performance been reached for all of the participants when linear drift amplitude stayed within 2 arc minutes, which is incredibly small eye motion. And then we actually investigated what will happen with the retinal space on drift semi-perimeter measurement. And it seems like we need 2 times smaller like space on a retina occupied to reach maximum performance, which is around 1 arc minute. And so to conclude, visual acuity performance improves with the higher cone density. Cone density strongly influences drift behavior, such as drift velocity and drift amplitude. Maximum acuity performance reaches the linear drift amplitude or 2 arc minutes, and space, which is occupied on the retina, seems not to be as that important as the linear drift amplitude. And of course, I want to thank you all my lab mates and colleagues, and our collaborator from the Lille University, and actually Julius and Alex, they're in the audience, so feel free to approach them and talk to them. Yeah.
It's open for questions.
Any questions?
I have a question.
Yeah.
Can you go on slide 19?
19? This one?
Yes. I know it's very hard to find patients for your experiments, but maybe it's a bit too much to draw a line between only 4 points for statistics.
Yeah, of course. I mean, here I said that I present data only for 4 participants, and ideally, of course, I would need more participants, and ideally some of them should be like in between. But so far we only have 4 participants. But like in regards to performance, actually Jenny, she has like quite a big population of participants, and we can clearly see that there is some relationship between threshold. But I agree, it's not like statistical line, it's just like to visualize, not linear regression or so.
So it's a very good talk.
Thank you. Yeah.
And concerning the distribution of the area when you see the drift, what's the size of that? Is it included in the fovea? Does it diverge to the less dense cone areas? I mean, it's,
Do you mean like for this participants?
for
Or?
instance, yeah, what is it relative to the lateral extension of the drift, and does it go into relative to the density of the receptors?
Yes. So
how is it drift in let's say in low population of cone? Does it go back to the high population?
Yes.
Like that?
Yes, that's exactly what happens. So for, I mean, it's again like Jenny like preparing her paper, but she showed that actually subjects that tend to start somewhere outside of the very foveal center, but then move stimulus towards the cone density centroid. For this particular subject, she's very interesting. So she's been always starting somewhere completely outside of the foveal center and then drag stimulus towards the cone density centroid. But we think that yeah, she will start like somewhere like here, for example, and then go towards. This is like normal for everybody, but for her, like it was extreme. Yes.
So you didn't find any acuity performance when stabilizing the letter E, but have you tried doing the same experiment but more in eccentricity, like far away from where the patient is staging?
Yes, so very good question, because actually like most of the work which been done with the stabilization technique been done outside of the fovea, and there we can clearly see the difference. In our case, we didn't do it on a periphery, but we only did it on a like very central part of the fovea. More specifically, we stabilized on the preferred retinal location. So this is the area of the fovea with which we actually fixate. So maybe that can explain our results, why we don't see any difference apart from like optical correcting for optical aberration stuff. Thank you.
right, our next speaker is Yao. Maybe save the I was going to say there's a folder, i2eye folder. This. Okay. So hello everyone, I'm Yao Cai and I'm from Paris Eye Imaging Group. So follow Veronika's very nice talk about the lateral eye motion. I'm going to talk about our recent study about the retina axis motion and how we can do for the real-time correction for the in vivo retina imaging. So as we know, when we perform the in vivo human retina imaging, there's a problem is the involuntary axial eye motion. So as we can see in this HD-OCT scan recording, the retina, it keeps moving during the image acquisition. And if we look at the HD-OCT M scan, we can see the very obvious oscillation and the drift along the Z direction, and it's within 5 seconds of the acquisition time. And this behavior is quite a major problem for the in vivo retina imaging by the optical coherence tomography, such as the time domain full field OCT. So what is the time domain full field OCT? I believe from previous talk, most of you have been aware of this technique. So we have the spatially incoherent light source from the AED. Then we have the beam splitter to split the light beam into the two arms. Then we have the backscatter light from these two arms and then recombined by the beam splitter and detected by the camera. So with this technique, we don't need to have the 2D scanning. We're less sensitive to the lateral motion. And we just need to move the coherence gate to get the volume imaging in the Z direction. And the signal intensity can be expressed by this formula, and it's composed of the incoherent signal and the coherent signal. So we can perform the two-phase demodulation to get the coherent signal and get rid of the incoherent signal. So how does this technique work for the in vivo retina imaging? As I mentioned previously, there is continuous involuntary axial motion. So we have the, for example, in the case of 8 microns coherence gate in the full field OCT, if we want to image the specific layer in the retina, we firstly can localize this coherence gate at the interesting layer, but then the eye can keep moving during the acquisition. So mostly it will get outside the coherence gate. Then we are not able to get the signal from this layer. So maybe we get the signal from other layer, and or we just get the noise. So what we can do to tackle this problem? So our strategy is to build a system, combine the time domain full field OCT and also the spectral domain OCT. So in the spectral domain OCT, we have the real-time B scan. We are able to have the we're able to monitor the axial position of the retina. So we do the computation by doing the cross correlation to measure the retina axial position in the real time. Then we move the reference mirror to match the optical path length. So this means with this strategy, we're able to localize the full field OCT coherence gate at the interested retina layer for imaging. So as shown in this video, and the coherence gate is marked by the black arrow. So we can move this coherence gate to follow the retina motion in Z direction. So how does it work in our for the in vivo retinal imaging? Firstly, I'd like to show the example in the eye cells layer. So we have the image close, full field OCT image close to the foveal center. And this is the one full field OCT image stack that acquired within 0.5 seconds. Then we do the image average, so we can have the MFAS full field OCT image with high resolution and over a large field of view of 5 degrees. And here we can see we are able to visualize the photoreceptor cells even down to 0.3 degrees from the fovea. So here I use the black star to mark the position of the fovea. So then we move to the nerve fiber layer. So thanks to the good optical sectioning of our system of 8 microns, so we are able to visualize the different details at the different depths of the nerve fiber layer. So here I show the example from the 3 layers in the nerve fiber layer. The first one is the inner limiting membrane, and it's on the top. And from this first image, we can see some bright spots, and the size is around 20 to 30 microns. And the second one in the middle, we can find some micro-sized hyperreflective spots along the axon bundles. And the third one is close to the third one on the bottom is close to the ganglion cells. And from this image, we can have a very nice view of the vessel wall and also very fine structure of the capillaries. And we can also measure the width of the vessel, and it's around 120 to 130 microns in this case. So thanks to this very accelerating result, we reinstalled our system in the Gansvan Ophthalmic National Hospital. And from yesterday's talk by Maxim, he showed the full field OCT is quite promising for the clinical applications. So we can have the high 3D resolution, and we can have a large field of view of 5 degrees. And the system can stay very compact of around 40 centimeters. And also thanks to the high frame rate camera, we are able to have the very fast acquisition. But the thing is that when we try to image broader population of the patient, sometimes we have very low signal to noise ratio. So we need to do the average of more image to improve the signal to noise ratio. And we need to have a longer acquisition of around a few seconds. And this is kind of challenging because of the continuous exit motion. So we need to have the better exit tracking. So here I would like to show the tracking performance from our system for the in vivo retinal imaging. The first case, this is the SDO-CTP scan from the one subject, and this healthy subject, and he get used to the eye imaging, so we can see the amplitude of the axis and motion is not very large, and we have a very good tracking. And from this plot, we can see the retinal position as shown in black, and the coherence gate of 8 microns is shown in blue. So we can see the black curve is mostly stay inside the blue. So it means the retina is mostly stay inside the coherence gate. We can have the tomographic signal from this layer. But on the other hand, there is some cases, so we don't, have a very high signal to noise ratio, high signal from the subject, and it can move fast with a very large amplitude. And in this case, we are not able to perform a very good tracking. Mostly the retina is outside the coherence gate. So in this case, in general, we can only have 10% of chance to get the tomographic signal with this not good tracking. So here we might need to ask ourselves if we can have a better axial tracking and how fast shall we go in terms of the axial tracking loop rates. So to answer this question, firstly, we'd like to have a better understanding of the axial motion. So firstly, I did some literature, and here I show the most representative the paper from Keynote published in 2011, and they studied the axial motion using the spectral domain OCT. And they have this is the plot of the axial motion in the time domain, and they also measured the blood pressure. So in the frequency domain, they find that the axial motion is linked to the respiration at 0.2 Hertz and also linked to the heartbeat at 1 Hertz. But as we can see, the scan rate of their system is not very fast. It's 5 kilohertz, and the axial resolution is around 15 microns. So to have a better characterization of the axial motion, we try to study with the higher temporal frequency, temporal rates of 800 kilohertz A scan rate and a better axial resolution of 4 microns. And also we studied over a large population of 14 subjects. And in this study, we also use this characterization to have a better design of the axial tracking. So this is the drawing of our setup to measure the axial motion. We have the spectral domain OCT, and we can get the SDOCT big scan within 3 seconds. And we want to investigate if we can benefit from the breath holding, because the respiration is one of the causes of the axial motion. So we ask the subject to keep normal breath or hold the breath. Then we acquire 2 groups of the data. So this is the plot to show the behavior of the axial motion in the time domain, and we can find this kind of oscillation and the drift in all the subjects. Then we do some statistics on this axial motion data, and we find the breath holding can help us to decrease a bit the amplitude of the axial motion. But it's very surprised to find that in terms of the axial moving speed, the breath holding seems to make the subject move faster. And this speed level at 1 millimeter per second is inconsistent with the figure that mentioned in Ginger's paper. So then I compute the power spectral density in the frequency domain, and then this is the average PSD among all 14 subjects, and the blue line is for the case of the breath holding, and the red line is for the case without breath holding. So here we can find a linear regression, and we can find dynamics of the axial motion up to 150 kilohertz, and then we reach the noise level, and this is because of the limit of the bandwidth. So firstly, we zoom in the region of the low frequency domain from below 1 Hertz. So here is the zoom view, and we can see the difference between the 2 cases with breath holding and without breath holding. And the difference is around a factor of 3. The case showing in blue, it seems to have a lower PSD of the axial motion. And this might be linked to the respiration or the body motion below 1 Hertz, because the breath holding can have the subject to have the respiration control, so we can reduce the axial motion in this frequency domain.
And then we move to the domain from 1 Hz to 2 Hz. And the difference between these two cases seems to be still very obvious. And if we look at this region, we can find the breath holding can reduce a bit the PSD of the axis motion. And from this paper, they mentioned that apnea can induce a reduction in the cardiac output. So we guess this difference, it might be linked to the heartbeat. And also this confirms that the axis motion in this frequency domain is linked to the heartbeat rate. And one interesting thing to share is that we find some peaks at 3 to 4 Hz, and this is not reported previously. Then if we take a look at the axis motion plot in the time domain, we can find very interesting behavior in this subject. And this plot is very similar to the plot showing the blood flow pathway. And this is also presented by Miguel yesterday. So we are wondering maybe this kind of peak is related to the high order harmonic of the heartbeat rate, or is it related to the pulsatile blood flow? And then we look at the frequency from 7 to 150 Hz of the high frequency domain. We find the breath holding seems to show a bit higher PSD of the axis motion, and this is not good for the real-time axis correction. And also, according to previous statistics, we find that the axis speed in case of the breath holding seems to be faster than keeping normal breath. So this means we might not need to have the breath holding for the in vivo retina imaging. And then after the characterization of the axis motion, we want to think about how to have a better design of the axial tracking loop rate. So here I simulate the axial tracking in a scheme of a 2-frame delay. And on the left, I plot the imaging success rate at different control loop rates. And here the proportion, it means the proportion of the data that we can have within the coherence gate, so we can have the tomographic signal from the imaging layer. And on the right, I show the distribution of the RMS tracking error among 14 subjects. So here, if we want to have the RMS error below 4 microns, we need to have 1.2 kilohertz to have the 100% of the good data. It means that we can have the robust acquisition. And this simulation result is also matched what they said in this purchase paper. They mentioned that they have the tracking loop rate of 1 kilohertz, and they have the residual tracking error is lower than 5 microns. And then if we can have a trade-off in the axis resolution, so if we want to have the RMS error below 8 microns, we need to have 600 Hz for the axial tracking loop rate, and it should be enough for the real-time correction. So one interesting finding in this study is that if we look at the dynamics of the axis motion in the time domain, we find one subject, it shows very regular and also high amplitude of the oscillation as shown in these plots. And this subject, the difference of this subject is that we perform the experiment after taking the coffee, and we can find a pronounced peak at around 4 Hz in the frequency domain. And from this paper, they mentioned that the caffeine can lead to the vasoconstriction. So we don't know if this kind of interesting behavior can be linked to the increase the blood pressure and also the reduce the retinal blood vessel area. And from another subject, we find this interesting behavior just after the breath holding. So as shown in this blue curve, and it's a uniform oscillation. So in this paper, they say the apnea can also introduce the vasoconstrictions. So we are thinking this might be also linked to the increase the blood pressure. So to conclude, in this study, we try to characterize the X-ray resolution with the high temporal and spatial resolution, and we proposed better design of the X-ray tracking, so we can have the 100% of the robust acquisition in the full field OCT for the in vivo retina imaging. And also for the OCT angiography, it needs to have the 3D HD-OCT measurement. So with a better X-ray tracking, we are able to have a more accurate measurement in the OCTA. And also we can reduce the complexity of the 3D image restoration. And in terms of the physiology, as I showed previously, some interesting findings have shown the X-ray motion, the amplitude, and the frequency seems to be linked to the blood pressure, the pulsatile blood flow. And from this paper, from the Leopold, they published in 2021, and they showed in the patient with high tension glaucoma, they find the abnormality in the blood flow. So we are wondering maybe the X motion can be a new biomarker for this kind of disease, and also it's the non-contact method, and it can be just a bonus from the OCT measurement. So thank you, and welcome to reach me out if you have any further discussions. Okay. Thank you.
It looks like there may be a question on Zoom.
Okay. Much the X motion might be related to the rotation of the eye. Actually, this is a very interesting question, because when we performed the experiment, we didn't consider the eye motion. So we have the fixation target to ask the subject to stare at the fixation target. Then we, when we measured the X motion in the HD-OCT. So maybe this might be interesting for the further investigation.
Any other questions? We'll thank your speaker again.
Thank you.
Out here. I'll let you open up. All right, our next speaker is Clara.
Hi, I'm Clara Pfeffler from the University of Lübeck, and yeah, I'm going to present the results of our ophthalmoscopy research group about optoretinography in the inner plexiform layer. And I'm going to start with a short introduction of the technique we are using, which is called phase-sensitive OCT. This basically means that we have to acquire our OCT data so fast that it's not that the phase of the backscattered light is not corrupted by any eye motion or measurement artifact. And we realize this by using a full field swap source OCT system, since we can here parallelize both lateral and axial acquisition. The lateral acquisition is parallelized by a collimated beam combined with an area camera, and the axial parallelization is realized by detecting not the depth profile itself, but its spectra. So we thereby generate phase lock volumes where the phase is no longer obscured by measurement or motion artifacts. So the phase change we actually measure contain information about morphological change of our samples with a nanometer accuracy. And we are using that to detect changes in activated neurons, and especially in the retina, since the retina has a very nice feature that is the layered structure, where the different cell classes are each situated in a very specific layer. The photoreceptor cells, bipolar cells, and the ganglion cells. Therefore, we can very easily investigate those different cell classes separately by just evaluating the phase change in the very specific depths of the retina. And this is basically what we have done in the past few years. We started by having a look in the photoreceptor outer segment. Here we stimulated our retina with an X-shaped pattern, and we observed that there's an increase of the optical path length in the photoreceptor cells, but we can also go into other depths of the same measurement, for example, here in the inner plexiform layer and the inner segment of the photoreceptor cells, or the outer plexiform layer. However, when we want to investigate the cells a little bit more detailed, it becomes more difficult because since we have a very short exposure time, we are not really able to resolve single bipolar or ganglion cells, which means we cannot assign our functional changes to a single cell body, which would be very interesting since there is not only one bipolar or ganglion cell type, but many different types which are sensitive to different features in our visual signal. And therefore, it would be nice if we could distinguish these different bipolar and ganglion cell types. And that's why we focused a little bit more intensely on the inner plexiform layer, because we saw here the opportunity to distinguish different ganglion and bipolar cell types without really the need of resolving single cell bodies. And that's because the inner plexiform layer is like the retina, a layered structure where specific bipolar and ganglion cell types, which are specific to a very specific feature in the visual signal, only stratify in very specific depths of the inner plexiform layer. This is shown here for two examples. The first one is the on and off stratification. So the off cells are in general activated by a light stimulus, and they tend to stratify in the inner layer of the inner plexiform layer, and the off cells, which are inactivated by a light stimulus, they tend to stratify in the outer layer of the inner plexiform layer. And the second feature shown here is that fast changes in the visual signal are more likely to be processed in the central layer of the inner plexiform layer, while slower changes are more likely to be processed in the innermost and outermost region of the inner plexiform layer. So in order to differentiate those different functional cell types, we just had a look at different depths of the inner plexiform layer, or the phase change in different depths. Basically, we were able to separate 3 different layers. Due to our axial resolution, we couldn't differentiate between more layers. And in the first step, we stimulated our retina with a continuous stimulation of 8 seconds. And what we observed here is that indeed the 3 different sublayers behave very differently to the same stimulation. We see that we have an increase in optical path lengths in the innermost layer, a decrease in the outermost layer, and virtually no response in the central layer. And that fits well to the previous description of the on-off cells, which react inversely to the same stimulation. And in the next step, we change our stimulation. We no longer use the continuous stimulation, but now we use the flicker stimulation with 10 Hertz. And what we observed here is that basically in the innermost and outermost layer, the signal pretty much stayed the same, but now we could observe a signal also in the central layer of the inner plexiform layer, which is sensitive to temporal change in the visual signal. So in addition to this axial signal processing, we also had a look on the lateral signal processing. And here of central importance is the center surround processing of the receptive field, which basically means that bipolar and ganglion cells are activated when they get stimulated in the center of the receptive field. However, they get inactivated when there's a stimulus in the surround area of the receptive field, which leads to a very strong response when only the center is activated, but a very low response of our cells if both the center and the surround is activated, which basically creates an edge detector of our visual signal. And to investigate that, we changed our stimulation pattern from an X to an square. And here we can see indeed very nicely that we have an increased signal response in the edge region of our simulation pattern and a significantly lower signal in the central region of our simulation pattern. And up to that point, we could show that we can investigate very different aspects of signal processing in the inner plexiform layer, but so far this was all done in one subject. So of course, the next step was to reproduce these findings and see if this is just the one-hit wonder or if it's really something which is reproducible in others. So we started with a small cohort of 6 subjects or volunteers, and we were especially interested in how the motion or the eye motion during the measurement affected the outcome of or the quality of our face evaluation. This is why for all measurement we tracked the motion in X, Y, and Z direction, and then we also compared 2 different algorithms for the face evaluation. The first one is a simple face referencing to a fixed starting volume. And the second one is the so-called extended Knox-Thompson algorithm, which is a statistical approach which is thought to be more robust in case of face decorrelation. And we first tested this for the signal in the photoreceptor cells, and as a criteria, we used the phase noise over time. And here we basically see that we don't have a huge difference between these 2 algorithms. And also the unflashed image here is the simple phase referencing, and this is the extended Knox-Thompson algorithm. Basically looked the same after 8 seconds. However, if we look at the functional signal in the inner plexiform layer, we see that right from the beginning, we have a significantly higher phase noise by using the simple phase referencing, while the extended Knox-Thompson algorithm provides us with a very low phase noise over the whole measurement time. And if we have a look at the on-path image, we basically see that we can no longer identify the functional signal with the simple phase referencing, which is why for the further investigation, we only used the Knox-Thompson algorithm. And could show that for the photoreceptor cells, we could, in all our measurement, detect the functional signals, and we had a high reproducible, internal reproducible within one subject with some outliers, which seem to be a little bit higher. When we go to the functional signal in the inner plexiform layer, we get a slightly lower success rate. Here we were able to measure the functions change in 86% of our measurement. but we could measure those changes in at least every subject once. And here, especially extreme lateral movements seem to be the problem to evaluate the face in the inner plexiform layer. And of course, we wanted to look if we could also see the axial tendency in the inner plexiform layer in other subjects. And indeed, we could see that we have a increase in the innermost layer, a decrease in the outermost layer, and a temporal sensitivity in the central layer of our subjects. Yeah, I think with that I'm already at my conclusion. What we showed here is that we distinguish different contribution of different functional cell types of bipolar or ganglion cell types in the inner plexiform layer by just evaluating the pace of different depths. We just did this for two different aspects, for on and off cells and for temporal cells which are sensitive to temporal changes. And of course, there are a lot of other cell types which have to be investigated in the future. So far, we only were able to show this for 3 different layers. However, we are currently trying to increase our axial resolution by combining 2 swab sources with each other. So hopefully we will have a more detailed investigation of that in the future. And in the end, we were able to reproduce those measurements in 6 subjects so far. However, we already got the ethic approval to do this in a pilot study with healthy subject in order to investigate or test our algorithm a little bit further and to identify potential clinical parameters. And with that, I like to thank everyone from the research group, especially Svea, which did the measurement, 6 volunteers, and of course the 6 volunteers I also like to thank. Yeah, and with that, I'm finished.
Question?
What motivates actually the choice of the frequency? Could you imagine you should increase the signal by changing the frequency of it?
Yeah, we did we had another project where we changed the frequency, and we saw that the highest response were when we used 10 Hertz, and before with lower frequency or with higher frequency, the signal decreases again. So that's why I showed here the 10 Hertz frequency.
It looks like you have a question on Zoom.
Oh. Yeah, I actually don't know what the physiological origin is. It was suggested that it might be neurovascular coupling, but I don't believe that it's actually what we see, because we don't see a lot of correlation between our signal coupling and the vessel system. But what exactly we are
I just want you to say the question before you answer, so they can.
Yeah. Okay. So I have no idea what the physiological origin is. I don't think that we, with our data, we can really say for sure what it is.
What is the reference?
Well, we.
Sorry,
we
read the question.
question is,
Oh,
so
okay.
that they know you're answering.
Okay, so the question is, what is the reference layer you use to subtract the phase of each other? And that's basically that a layer is reference to the layer exactly below that. So the exact 1 pixel below the one layer is reference to the layer 1 pixel below.
Any other questions? Right. Thank you.
Let you get your talk ready. Our next speaker is Slawomir.
Yes.
Did I say you? I hope. Yeah. Thank you.
Bottom on screen.
Oh yeah. Yes, so hi everyone, I'm happy to be here, and I'll be talking. My name is Slawomir Tomczewski from i2eye Poland, and I will be talking about optoretinography with use of flickering stimulus. So, for many years, to have proper diagnosis, and to detect prevent vision loss, we need to have methods for early disease detection and tracking progress of the therapy. And for many years, visual inspection of, for example, OCT or SLO images or fundus photography has been used for these purposes, but it sometimes might be hard to spot the changes in the retina at the beginning of the disease. And for that, functional information might be helpful in some occasions. So there are multiple ways to obtain functional information. So there is psychophysics, which is subjective, and under these measurements, the person is asked if she sees some phenomena, for example, flickering light, and the intensity of this flicker is changed up and down until the threshold is being found. There is electroencephalography, so measurement of the brain waves. And here electrodes are mounted around the subject's head, and for example, the person observes the flicker and the amplitude of the signal is detected. So here is an example from literature. We have a different frequency of the stimulus and different different responses at these different frequencies of the stimulus, and amplitude decreases with increase of the frequency. And then there is electroretinography, which has been developed for many years. And in this method, electrodes are mounted usually on the cornea and in proximity to the eyeball. And in this approach, the changes in electric current or potential is being measured. These experiments have been done in flicker mode, in light-adapted conditions, or in dark-adapted conditions with a single pulse stimulation. And finally, there is optoretinography, which have been presented in talks during this session. So it was summarized by Ravi Journal as a means of measurement of light-evoked changes in the optical properties of retinal neurons. And the history can be given us, there have been multiple approaches to this method. So one of the common one is OCT. So in 2005, 2006, 3 groups worked on detecting observed changes in the reflectivity of retinal rays in animals in vitro and in vivo in OCT systems. Then other approaches were also used, not only OCT. So this is a simplified example. When we shine the light at the retina, there is some change in reflectivity or in a segmental to segment junction. And this signal can be quantified, for example, by measuring the amplitude of the change of this intensity, or can be also in interferometric methods such as OCT can be quantified as measuring the change in the phase of the phase difference between retinal layers. So there have been multiple works on this subject, so I could put some of them. I was not able to fit all, of course, but most of them were focused on the single pulse stimulation. And when we were starting our work, when we looked at the periodic stimulation, we only found 3 works. So one was by Tishomo et al. From 2010, sorry, and it used the scanning CT system and focused on measuring the amplitude. So the retina was stimulated with 5 Hz stimulus, and then signal in each pixel was Fourier transformed in time, and amplitude of the changes at the frequency of the stimulus was displayed. So this is visible in a segment, outer segment junction. And then there were 2 more recent works from Lubeck, some from Clara, who presented previously, and one was similar to what we are doing now. So measuring the amplitude of oscillation between inner segment, outer segment junction and outer segment tips, and the other one was focused on the inner plexiform layer and measuring the response there. So in our work, we use full filter CT system with a swept source laser. Then there is a multimode fiber here because a swept source CT system suffer from lack of the mechanical pinhole, so there is a crosstalk in the system. And Egidus came up with a way that using this multimode fiber decreases the spatial coherence and actually reduces the crosstalk in the setup. When this light emit goes from this optical fiber, it hits this rod mirror, which I will explain in a few seconds, and then it goes into the interferometer when it's divided in measurement and sample arm, and then its interference is registered on this camera. And the issue with this camera is that it allows for really fast data acquisition, but slow preview because it has to switch between acquisition and transfer data mode. So we get like 1-2 frames per second with the preview, and it's really difficult to align the subject. So to avoid this problem, there is a preview camera and the setup is a little bit tilted. So the light that would be otherwise lost is reflected in this direction and registered on this line scan camera, which allows about 30 frames per second, a little bit more. In addition to that, we have a stimulus path. So this is white light LED from Torlabs. We have DMD, which allows us to project the patterns. It's conjugated with the retina surface. And we, as I mentioned, we track the phase between a segment, also a segment junction, and outer segment tips. So first, we try to replicate results done by our other groups. So single pulse stimulation, we were able to observe contraction at the beginning and then expansion in the phase difference. And this is average from 3 measurements. Then we did some patterns. So this is checkerboard. This is letter E, which we were also able to recover. So the area of measurement is about 1.7 millimeters at the retina to 0.85 vertically. And to process our interferograms, we use the following scheme. So first we have to acquire data. So we acquire volume within 5 milliseconds. And when this volume is acquired, we swap laser in this spectral range. And we continue that volume after volume. While doing that, we stimulate retina with single pulse or multiple pulses, depending on experiments. Then we have to generate complex OCT volumes. So this is done by windowing, filtering, and then we correct for in-sweep transverse movement, because sometimes we observe that the volumes were corrupted if the rapid movement of the eye occurred in transverse direction. So we divide spectrum into subspectrums. We compute the lower resolution volumes, and then we track the direction of the movement, and we correct for that. And then we perform volume alignment, rotation removal, and increase accuracy volume alignment. So we apply padding to that volumes before computing the correlation. So then we use a simple flattening. So we axially shift each A scan to have RP at single level, and then we manually mark the regions in which we suppose the layers of interest are present. Then we perform volume referencing to cancel arbitrary phase offset, and we extract the phase difference between inner segment, outer segment, and outer segment tips. Finally, we high-pass filter signal to remove low frequencies, and we Fourier transform it to display the spectrum. So we checked 2 illumination scenarios. One was close to dark adapted, and by close to dark adapted, I mean that we have some fixation target on this DMD. And so we have a little light to have a proper position. And this light leads to about 7% breach of the photoreceptors. And when we use these scenarios, we notice that when we started flickering with increased intensity, we observe this combination of something that looks like this high amplitude changes for dark adapted scenarios with some oscillations visible here on top. And we had this problem that in this region, we could not recover our response because we've been seeing a lot of noise. So we checked another scenario in which we pre-illuminate the retina approximately with the half of the intensity of the flicker. This was leading to 81% bleach. And then we observed only these oscillations between the measurement layers. And this is our signal average from the whole point of view, field of view. So here I'd like to thank Robert Zawadzki for helping us to understand how to calculate bleach, because we had no experience with that previously. And then we performed such measurement under different cases, so 5, 10 hertz, and so on. Of course, the dark line is the average, and the light blue is the standard deviation of the measurement result. And from that measurement, we reconstructed the phase difference for these two layers. So inner segment, outer segment, and master segment tips. And this was one of our first results to compute that. It was computed from 50 measurements, so a lot, and it took a lot of time, I would say. Then we tried to check if we can actually separate these responses spatially. So we used the DMD to project stripes. First we projected vertical stripes of 15 hertz separated by an area that was not flickering. And then we analyzed signal in each pixel separately, and we bandpass filter the response around the frequency of the stimulus, and we display the amplitude. So we nicely recovered the responses in the areas that were illuminated. Here we had some problems due to vignetting in the measurement. And then we tried a little bit more complex approach, so separate responses from 15 and 18 hertz. So this is with Riemann measurements, we acquired a set of volumes, and again we analyze signal in each pixel, and when we bandpass filter the signals in each pixel around 15 hertz, we saw a response in one area, and when we bandpass filter the response in the other range, we saw the response from the 18 hertz stimulus. And after these experiments, we tried to improve the how to make those measurements in convenient way and don't repeat this 50 times for the characteristics. So we tried chirp flicker stimulation. In this simulation, we used the similar parameters. So 5 nasal to the fovea, one light adapted for 1 minute, and some similar bleach, but instead of a single frequency, we applied 5 to 45 hertz stimulus. So something similar to this. And for this measurement, we decreased the area to do camera memory limitation, and we increased also the number of volumes by a factor of 2. And we get responses as this one. So this is after high-pass filtering for a better presentation. And this is average from 5 measurements. And from such measurements, we compute spectrograms, from which which was short time Fourier transform, from which we are able to extract the amplitude of the response. So, for example, we know that in this time window, we have average stimulus frequency about 7.5 hertz. So in this time window, we reach response at the 7.5 hertz, and so on. And from such spectrograms, we can reconstruct the curve for the response. And this is comparison. Blue one is the response measured at a single frequency measurements, so 50 measurements, around 50 measurements, and the red one is the response acquired with the chirp stimulation. So the time it was only 5 measurements, then there is nice overlap. We have to investigate it a little bit more deeply because this is early results, but it seems that it looks really promising for a fast way to measure these characteristics in this region. And finally, we started measuring healthy subjects, so volunteers from the labs, from the lab, and we did this experiment on 7, but for some cases we changed the parameters. So I will display 3 of these measurements under the same conditions. So these are 3 subjects, and there is some difference visible, but I wouldn't go too deeply into that because we have in the amplitude, because we have to do more measurements to actually confirm that really there's a difference, but it seems similar to what was presented at the first presentation, that for some subject the amplitude differ. And finally, to summarize our talk, we demonstrated that we can detect response in quite repeatable fashion. We showed that we can measure response at higher frequencies, and we can detect response to pattern flicker. And probably most importantly, we showed that we also can use a chirp for the faster characterization of the retinal response. And this is at higher the prospect for novel retinal function studies and so on. But I have to hold on this because now we measured only the samples that are ready to measure. So volunteers from the lab who know how to focus, know how to maintain proper position, so they give us the stable signal. And now we'll be trying to switch to other subject and check if we can do also this measurement in a proper way, and we can get proper signals. And I would like to thank all the people here that worked on this project and other people that also worked on the setup and were really helpful in the development of the method. And thank you very much.
We're open for questions. It looks like there may be one on Zoom.
Okay.
And if you could just read it out, that would be great.
Okay, we'll go back to that slide maybe. So what's the reason for 2 peaks in the power spectrum measurements? And okay, so the peaks here on the left are just the results for remaining filtering of the signal, and peaks here, the peak here might be second harmonic, we are not sure, and this is for sure also remaining of filtering. So we had some uncorrected movement on something that gave or changes in the retina due to blood flow that gave some low frequency changes that were not completely filtered from the recorded signal.
Maybe as a curiosity, it was a very nice talk. I saw that your precision is almost nanometer. The measurements really show the. Can you comment on it?
Yeah, we were considering that and thinking about it, and we in the paper we actually calculated, and it's approximately as it is here. But the reason is because here we average the signal also from the multiple pixels. When we try to do this with this patterned one, the signal gets worse. And the issue, other thing is that we also use this Fourier transform, so we have like a lot of volumes from which we compute the spectrum. So it's nicely uniformly propagates across the high frequencies. Okay, thank you very much.
Get out of here. Sorry. Our final speaker of the session is Laura, and I'll let you take it away.
Great, thanks. So I'm Laura Young from Newcastle University, and I'm going to tell you about some work we've been doing to understand how accurate an ASLO is for eye tracking. So really understanding what level of accuracy can we achieve, what the main sources of error are, and how those propagate into standard metrics of fixational eye movements. So I presented some components of this before. I'm just going to focus on 2 aspects of it today. So firstly, just a quick view. So Veronika talked about fixational eye movements earlier today. And there are three main types of fixational eye movements: microsaccades, which are small ballistic movements; tremor, which is a small amplitude, high frequency oscillation; and drift, which is more like a random walk. And these carry the image over the retina of the order of the diameter of a few cones. And it's been known for a long time that this helps to counteract visual fading. So by moving the stimulus, you create a spatiotemporal signal, which helps to keep the photoreceptor signal from decreasing due to adaptation. And by translating the spatial information into a spatiotemporal structure, that can actually be beneficial to vision. And some studies have in fact shown that this can actually be useful in terms of visual acuity. But we also know that fixation instability is a feature of many ophthalmic and neurological diseases. So, for example, in central vision loss, fixation becomes highly unstable. But also in neurological diseases such as Parkinson's, we know that eye movements can be disrupted. And also in disorders such as concussion as well, eye movements can be disrupted. So it's kind of along this general theme that we've seen of using the eye as a window to the health of the central nervous system. This is another metric that we can use to kind of get at that sort of information. So I've put a selection of references here, but I just wanted to highlight that these ideas have been around for a very long time. So almost 100 years, people have been thinking about fixational eye movements and what they do to the retinal input. But it's only recently that this type of research has really had a big influx from developments in the technology. So specifically, the technology that we use is adaptive optic scanning laser ophthalmoscopes, which we've heard a bit about yesterday and today. So I won't go into too much detail, but essentially this is a scanning system. So the image is built up pixel by pixel over time. And you can see an example image on the right-hand side there. So as we build this image up pixel by pixel, we get these rolling shutter distortions. So as the eye moves during the scan, we introduce the distortion, and you can imagine that the direction of the eye movement and the speed affects how that's distorted. So for example, if your eye moves very quickly upwards against a slow scanner, you'd get a severe compression of the image and you start to lose bits of the image. Here you can see an example where the eyes move sideways and you can see this kind of stretching in one direction. So what we want to do is estimate what these distortions are and use that as our measure of eye movements. And I'm going to talk about two different ways to do that. So the classic way that people have been doing this is to use strip-based cross-correlation. So originally suggested by Jeff Mulligan in 1997, and it's had a few implementations, most recently by Zhang from Ethan Rossi's lab in 2021, of using strips of the image that you cross-correlate between the frame that you want to make the measurement in and a reference frame that you choose or that you construct to have no motion or minimal motion in it. And the parameters that you can select here are the size of the strip and how many strips that you take down the image. So you can, for example, you could overlap those strips to get more data samples. You also, when you do this, you generate your normalized cross correlation, and the size of the peak of that normalized cross correlation tells you how good the match is. So you have a metric that you can use to reject bad data. Now, what I'm going to talk about now, I haven't done any of that rejection, let's say that now. That's another parameter that I will be testing. The alternative approach is one that we've used in our lab, which is to use a dense optical flow algorithm, so the Farnbach optical flow algorithm. And what this does is it looks between two pairs of frames on a pixel by pixel basis for motion. And it does that by fitting a polynomial to the intensity pattern and then looking at how that shifts one frame to the next. So I've drawn some arrows on that diagram, but actually you do get this measurement per pixel. And again, this has a certain set of parameters that you can tune. So it fits the polynomial over a certain window size, so you can change the size of that window. And it also does this over certain scales as well. And so as you adjust these parameters, you're kind of trading off accuracy and precision for small eye movements versus your ability to detect larger eye movements. And so just to show you, here's an example trace from an AOS alone. This was the system we developed in Oxford. And if we compare that to the standard video-based eye tracker that people use, which is the iLink 1000, you can see a clear difference in terms of the precision of the measurement. So there's about an order of magnitude difference, at least, between these two. And in fact, you can see around just after 1 second in the AOSLO trace, there's a clear microsaccade that's a bit more difficult to detect in the iLink. So clearly the precision is a lot higher, but if we're really going to claim that we're measuring things to single cone accuracy, we'd better be sure that what we're actually measuring is coming from the eye itself and not through something that we're doing in our data capture. So something in the system itself or something in the data processing pipeline. So we want to look at how accurate this is. We need to think about what the sources of error are. So firstly, there might be effects from the bench itself. So for example, it could be vibration through the bench. You could have the beam wandering due to turbulence on the bench. There could be electrical noise that's affecting the position of the scanner. And you could also have effects from the AO system if it's become unstable or if you've got unsensed tip tilt. You've also got the conversion between your intensity signals that come out of your detector and that converting that into an image. And so that might be new electronics to do with synchronization and timing. And it might also be to do with whatever desinusoiding algorithm that you use. You've then got these set of images. So then you have to extract the motion. Talked about 2 ways of doing that. So depending on which way you do that, which reference frame you select and how you set up those parameters, those can all have effects that might affect your accuracy. And then finally, it's what you do with the data afterwards. So do you remove microsaccades effectively? Do you remove blinks effectively? How do you characterize the movement? So I'm not going to talk about image reconstruction because everyone's system is slightly different and I don't really want to tackle everybody's different data capture system. And I'm also not going to talk about the eye movement trace analysis today because that is sort of common to any type of eye movement data you might get out. So first, just to have a quick look at bench effects. So here's the system we're building in Newcastle. It's almost online. We've got an AO system just about up and running, but we haven't quite had first light. And this has been built by Penny Lawton at the moment. And this is just on a standard Full Labs honeycomb breadboard, 110 mil thick with passive isolation on the frame. And what Penny's done is just to introduce a model eye, so something that's not moving, into the system and see what we measure. And here's an example model eye movement trace showing the horizontal and vertical movements. And you can see that the vertical movement is a lot noisier than the horizontal movement, but the RMS error is still under 0.12, sorry, arc minutes RMS, which is absolutely tiny. It's about 1/50th of a cone diameter. So it's really small. But if you look at the power spectrum, sorry, the amplitude spectrum on the bottom, you can see that that's all concentrated in a few frequencies, particularly for the vertical scanner. And so that is actually the mains frequency in the UK. So that 50 Hertz peak is coming from electrical noise on our vertical scanner. And there's also a 100 Hertz signal, which comes from the AC to DC conversion through the rectification process, and then a bunch of harmonics. But these are generally very small, so we're not too worried about these. And we can actually measure these simultaneously and remove them if we need to. We also then repeated this with someone sat at the bench, sort of pressing on it a little bit just to see if we get in a patient that's perhaps not behaving very well, what the difference might be. And you can see there's not much difference at all. The RMS error goes up by a very small amount, and it's all concentrated in a little 5 Hertz peak down here. So we're not too worried that vibration effects on the bench are going to be measured, and we're going to make some kind of incorrect inference about eye movements based on that. Okay, so next we'll look at the way that we actually analyze the data on the frames that come out. So what we're going to do is use Erica, emulated metal image capture, to generate some synthetic images. So this gives us our ground truth data for the eye movements. We then use these algorithms to extract the motion, and then we compare the extracted motion via a set of standard metrics for eye movements. So firstly, Erica generates realistic AOSLO data streams. So it has a model of the retina. This is an analytical model, so we don't get sampling effects as we scan. We then introduce a series of eye movements that we add into the scanning system to simulate the movements of the eye relative to the scanner. And then we simulate the frame generation and data capture process. So that includes diffraction, noise, the pinhole, the desinusoiding process, all of those sorts of things. And we can add noise and aberrations to those if we want to. If you're interested in using this, you can find it on GitHub, or you can drop me an email. Okay, so we've got these synthetic images. You can see two examples on the right-hand side. So one of the fovea. So you can see, again, the cones are smallest and most concentrated in the center, and you can see them becoming more widely spaced away from the foveal center. And another example at 2 degrees. So typically when we're doing our eye movement studies, we might not necessarily image at the point where we're presenting the stimulus if we don't want that AOSLO beam to be visible. Or you may be introducing your stimulus through the AO system, at which point you would direct the beam to the fovea. And what we've done is to introduce eye movements just for the drift component. So I've done a separate study of microsaccades which have their own set of problems. So we've isolated out just the drift components. And that's been modeled as just a random walk. And what I've done is choose different step sizes for that random walk. So a typical sort of healthy person, in the way we've set this up, would have a step size of around 0.1 arc minutes, something like that. So this is someone who's fixating very stably, and this is someone who's fixating very unstably. So it might be more like someone who's got some kind of eye movement disorder. It's a relatively long duration. And the reason for that is we want the eye to drift far enough. Normally that would be achieved through a microsaccade. So we just run the random walk a little bit longer so we kind of reach that extent. There are 10 repeats, as I've said, 2 eccentricities, and the system is set up for a sort of standard AOSLO imaging condition. So 512 by 512 pixels, 0.1 arc minutes per pixel, a wavelength of 840 nanometers, 2 airy disc diameter pinhole, and a 7mm pupil diameter. Hopefully the movies play and you can see kind of what that looks like. I've also added some noise into these frames sort of based on standard images that we've collected. So you can sort of see what these look like. Okay, so next we've taken those series of frames and we've extracted them using 2 different algorithms that I've previously explained. So the first is the one from Min Zhang in Ethan Rossi's lab, which I believe they've renamed Remedy. And in their original paper, they stated that they didn't implement subpixel registration because when you're generating an image, it doesn't really gain you very much. But for eye movements, you might perhaps want to add that in. So thanks to them for sending me the MATLAB file to allow me to add that subpixel registration in so that I can make this a really fair test between the two. I've set this up with 16 row strip size. 512 strips, so these are overlapping, so it's moved gradually down by 1 row. And they also generate their own synthetic reference frame, which is supposed to be the kind of best reference frame that you can use to compare these two. And then for the optical flow algorithm, you don't have a strip size, it gives you 1 per pixel, but I have chosen to use either a randomly selected single frame, so this might be the best frame that you would select from your stream, a motion-free montage. So this is the ground truth. You know, an area that's the frames are going to not walk outside of, but also do not contain motion. And then also the reference frame that's been created by Remedy. And I'm going to compare these through a number of different metrics that you might use for parameterizing fixational eye movements, particularly drift. So firstly, just the RMS error, the bivariate contour ellipse area, drift velocity, drift amplitude, and a random walk parameter. And I'm just going to explain what that random walk parameter is because it's a little bit less standard, so you might not have come across it before. So this is some work that Ali Hexley has been doing at Oxford. And if you take a random walk and you plot on a log-log plot, the lag versus the mean squared displacement between those 2 time samples, you should get for a random walk a straight line with a gradient of 1. But if you try other types of random walk, so SAW is a self-avoiding random walk, WIMP is a walker in a moving potential. So this is a random walk confined to a potential, but that potential is also moving around. You get characteristic different shapes in this log plot. And so these parameters tell you not just about the amount of movement, but just something about the type of movement that's happening, or something about the behavior of the eye. Okay, so first the RMS error. So this is just the RMS difference between the ground truth and what's been extracted. And we can see that as we increase the step size, the RMS error starts to increase. And you can see when you select just a single frame, random frame, very quickly, the RMS error starts to increase. And that's because the eye movement has walked off that reference frame. So we'll just exclude that one so we can look in more detail at the others. And you can see that both the optical flow and remedy perform very well. So their RMS error is below the size of a foveal cone. And you can see that the optical flow algorithm performs ever so slightly better. However, and it's pretty much the same between the horizontal and vertical. However, if you extend that out to those more extreme eye movements where it's very unstable, the optical flow algorithm starts to break down. And this is probably because the parameters aren't really tuned to these large movements. So remedy is more robust to these less stable movements. And the same in the horizontal direction. Let's take some water, sorry. Okay, so bivariate contour ellipse area. So this is just an ellipse incorporating 68% of the data points. And you can see that, again, if you use a random frame, there's much more variability. So these error bars are one standard deviation. This is plotting the BCA that we've measured versus the BCA that went in. So ideally we want this to be 1. And you can see that as the step size increases, we start to make errors in that measurement. But if we look at the two algorithms with this synthetic reference frame generated by remedy, they're actually both very good at reconstructing the BCEA that it should have done. And the next is drift velocity. So it's just the gradient fitted to that drift profile. And again, they all do a pretty good job of reproducing the velocity that's gone in, again, apart from the single frame measurement. So if we zoom in on the two that use the synthetic reference frame, they're both hovering around about 1. Next is amplitude, so this is just the extent of the movement. And again, the same pattern that the single frame tends to have a slightly worse error in terms of measuring the amplitude that went in. So we'll remove that one. And you can see that remedy slightly overestimates the amplitude for small random walk step sizes. Now, again, I haven't optimized a cutoff in terms of the metric for how well these have matched. And I imagine if I did that, then these errors would perhaps come down a little bit. And then finally, the random walk parameter. So how well does it fit to a straight line? And a straight line with a gradient of 1. So again, the random frame, as you increase the step size, the gradient starts to change dramatically. So you might infer that it's not actually behaving like a random walk anymore. And the two using the remedy synthetic frame both perform very well in terms of reconstructing a gradient of approximately 1. So if we look at the R squared, so how well does it fit to a straight line? Again, the two that use the synthetic reference frame very closely fit to a straight line. So that's great. It shows that if you use these two algorithms with the remedy generated synthetic reference frame, you're going to make correct inferences about the drift movement. So just to summarize, if you use a single frame, it has the potential to cause large errors, particularly when you've got large movements that sort of walk out of that field of view. Remedy is also really good for generating a reference frame that's very close to the ground truth. So you can see that it gets very close to this motion-free condition, which is the best that we could do. And again, remedy has a slightly larger error, but is actually more robust to these larger eye movements. So considering that they're both very accurate, maybe that's a good trade-off to make. Okay, so just to sum up, we were slightly worried about bench effects, but we've been reassured that actually these are very small and unlikely to infect any inferences we might make about the eye movements that we're measuring. A single reference frame can introduce large errors, so if you can avoid it, that's best. Remedy produces a very good synthetic reference frame, which is a very good approximation to the ground truth, and both methods perform well and tend to produce drift parameters that are close to the ground truth. Remedy is more robust to large eye movements, but at the expense of small, slightly less good accuracy at the more stable movements. So finally, just to acknowledge my team. So this is a relatively new team. So Stephanie, Penny and Veronika all joined the lab within the last year. And also to thank the team in Oxford, particularly Ali Hexley, Hannah Smith and their collaborator Tom McLeish, who've been working on the eye movement parameterization. And thank you all. Take any questions.
So sure, Erica, mosaic simulated images. Can you create a variety of images that would model different densities, and are they like they're newly generated each time? Do you put the input parameters of density or eye length or things like that?
Yeah, so the actual position of the cones is generated from a reaction diffusion model. So it's a sort of a random pattern that's pseudo-regular. So you can adjust that and make it more or less regular if you want to. And that's scaled to match histology. So it's concentrated in the center and expands out. So if you wanted to, you could adjust those parameters and change that scale factor with eccentricity if you wanted to. It also models the size of the cones based on histology as well, so you could adjust that parameter. You can change the histogram of the reflectance profiles. It's tried to make it as sort of adaptable as I could foresee anyone wanting to adapt it. Yeah, and you can also have them vary in time as well, so you can have the reflectance changing over time.
Do you want to check if there's a sorry.
Yeah, there's also one over there.
Yeah, I wanted to ask you, why did you choose these parameters, like for random walk? Because I think from frame to frame, if it's measured only on the cross, like from frame to frame, now I saw system, it usually takes for participants like half a full diameter at least. So like 15 seconds. Yeah, why did you choose those parameters for random walk, different intervals?
Yeah, okay, so the question was why did I select those particular step sizes for the random walk model? So that's just based on a set of data that we've collected from normal participants, where Ali has modeled that log-log plot using different step sizes for a random walker, and then found the one that best fits the data that we've collected from normal observers. And so we found that that was about 0.1 arc minutes if your step sizes are 1 millisecond. And then I've just selected a few around that just to kind of gauge more or less stability or faster or slower eye movements.
Thanks. So if we could just thank all of the speakers in the session again. That was a great first session for today. Thank you all for your attention. How quick on the break?
We probably want to take a quick break. We have some coffee, etc. That's just here. I'll get that out, and we'll maybe meet back in 20 minutes, just after 5. For the next session.
Sounds great. All right. See you all at 5 o'clock.
Yeah.
So for those who are on Zoom, if you didn't hear that, we're going to break for 15 minutes and reconvene at 5 o'clock Paris time. All right, thank you.